Okay, good morning, everyone. It's grateful for the Gairdner Foundation to give me this prestigious award. Today, I would like to highlight those works conducted in our institute. In the last 2 decades, the first 2 decades of the 21st century, there have many important EID emerging in this global. Many of them is actually originated from China in our region. This include the SARS and the hyper H5N1 and the H7N9 bird flu, and most recently and the ongoing COVID-19. And among this list, most of the important events our institute directly involved and provide critical information to understand how this disease is emerging. Our missions of this institute is first to conduct pandemic influenza preparedness in Asia. That include identify those viruses with pandemic potential, prepare vaccine and diagnostic reagent. Since the SARS outbreaks, we found we have another mission, is understanding the emerging pathway of novel virus in this region. Why China? China, in the last centuries, we have two times, 1957, 1968 pandemic, and 1977 pandemic, all emerging in the Southern China. So this region was conducted as an epicenter of influenza virus for emerging pandemic flu. In China, we have the most comprehensive, complicated influenza ecosystem because we have the largest human population and animal population. We have the largest number of poultry, birds, and also largest number of pig. Certainly, largest population size of human being. Importantly, we have this type live poultry market system almost in every city. So these fact interactions compose of the most comprehensive influenza ecosystems, give the virus a lot of opportunity. So in the last 2 decades, this include the Southern China, where weekly sample more than 6 provinces, and also collaboration with the Southeast Asia country. Over 2 million of animal was sampled, and over 140,000 virus were isolated and identified. We sampled the animal from a retail market, Sentinel farm, migrating birds, hospital, even the slaughterhouse. When the bird flu H5N1 outbreaks in Hong Kong, we are the first team to identify it. And after the system set up, all the H5N1 variant was identified from our institute. And this includes V1, V2, V3. Those viruses change year by year, region by region. This is the institute to identify how and where this virus emerged, and how this virus transmit, even spread out across the board boundary. Go to the Southeast Asia, to North, go to Europe, to Africa, even eventually across the Bering Strait to the North America. We know this virus from time to time jump to human and cause the human severe disease. So this virus still in the field and still pose a significant pandemic threat to us. H9N2. This is another lineage, major influenza lineage in the poultry. I working on this virus since 1996. It emerged in 1994. Currently, H9N2 virus lineage is highly prevalent in the chicken and other poultry. And from time to time, this virus go to the human, jump to human. So when we surveillance in the human population, we found a high seroconvert rate in the human. If we calculate it, there are millions of people exposed to this virus. And like H5N1 lineage, the H9N2 virus also reassorted with other virus, generate different genotype from time to time. So far, this is still another major pandemic threat virus. So we have a systematically swine influenza surveillance network. So in the year 2009, when the swine flu emerged in April, our institute is the first one with 1 week time, based on our surveillance data, we understand this virus emerging pathway. Even this virus first emerged from Mexico. But its gene segment is actually come from a different continental. China, because we import the live pigs, both from a European country and American. So in this land, we have all components of this pandemic virus. So initially, many colleagues in this world think this pandemic may be initiated or originated, generated in China. Now it's until the year 2014, we eventually know this virus, it is happened in Mexico, okay, initially. Since the year 2009 pandemic emerged, the virus actually get back to the peak again, and significantly change the landscape of genetic diversity of swine influenza virus. I highlight here all colorful. This is the new lineage of swine influenza virus. From this fig, we know how the European virus and when it invaded China, how American virus in the early year 2002 get into China. So this land still have a most complicated swine influenza virus. We don't know among these variants of swine flu, which one will be cause trouble again, give rise to another pandemic. So our work is keep going on. Year 2013, in the East China, Shanghai, and reported to have a human case infected by H7N9. Based on our systematic surveillance, we very soon identify how this virus emerged and generated, both from migratory birds and also the chicken virus. And the pathway is clear, demonstrated in this picture. Okay. This virus actually is more severe than H5N1 and the other virus. Even initially it's low path, it is still infected a lot of people, caused the severe pneumonia. And this is a province by province, region by region, how this virus is way by way, season by season, expand that territory and almost transmitted to across the whole China. Okay. This is show you the high path variant, okay, in fact more than 15 provinces. Based on WHO data, more than 1,600 people infected or identified. But the mortality rate is as high as 38%. So this is not a friendly virus. We can assume if this virus really caused a pandemic, that's big trouble. It's disaster for the human being. Currently, by vaccinations, seeing that this virus is a slowdown to transmit in the field. But we have evidence the virus may come back. Now I change my topic, go to the SARS and the SARS-related, the emerging and the re-emergence of SARS coronavirus. We know, actually, based on the influenza surveillance and study, we understand the domestic animal is like our human beings safeguard. Any disease attacked us, they initially will come out from those domestic animals. But this concept was changed by SARS. Okay. Suppose those wildlife, if they want to get into human, they must replicate, attack this domestic animal first, and then go further to our human being. One example is some vectors virus by mosquito taken from any kind of animal, go to human directly. SARS. In the year 2003, and the SARS was identified, was caused by SARS coronavirus in March. And this gives us the right way to diagnosis and control. But another very important question is, where this virus emerge, how they attack human. Okay, we investigate the live animal market because many of the index cases of SARS, they have a history, contact or indirect contact with the wild animal. Because China developed gradually, the requirement for the food is increased. So we enjoy the exotic animal, like a civet cat, like a cool dog. This is a mammal. Okay. In the Zhejiang Delta region, this is the sales on the live animal market. So on May 8 to 9, year 2003, I visited the Dongmen market and the sampling there. we successfully identify this virus. First, this is a human SARS virus. Okay. This is Guan. It's the first human identification SARS. Sampled year 2003, February 10th. These are the two viruses I isolated sampling in February 17. This virus we sampled on May 8, all from Shenzhen. In comparison with the human virus, they become a sister group. They are not included inside. Genomically, we found the gene organization changed. The animal virus have 29 nucleotide gene longer than the human virus. Among these human virus, only one virus within this 29 nucleotide is Guan. That means based on the evolution theory, we understand the virus after jumping into human, they lose this section. So that's more directly, very strong evidence. The virus is transmitted from an animal go to human side, not the other way around. Okay. So we basically understand how human SARS come from. Then later on, somebody don't believe. They still not ban, they left the ban for the sale, those wildlife in the live animal market. In October year 2003, those animals come back. Okay. We quietly start reinitiating the surveillance in the end of October. Okay. The sampling period from late October to the January 1st of the new year of the year 2004. We totally sample 179 animals. It includes these 5 different species. Like civet cat, we sample 44, but 32 of them, about 72% animal carry SARS. And this is the number, detail. Okay, so I understand SARS is come back. At this time, to remember, we still don't know where the SARS come before this animal. It's until year 2005 or 6, people's report, first by another Hong Kong U team, Dr. K.Y., Yuan, and also later on in the Wuhan, Dr. Shi Zhenli's teams. That means the natural reservoir from bats, they have the SARS-like virus. Okay. But this happened in the late year 2003 and early year 2004. So we conduct final genetic analysis for those new viruses we detected in the late year 2003. What we found? This is the new virus come out after the October year 2003. This lineage is the May we detect in the animal, in the SARS, in the live poultry market, live animal market. This is the human SARS virus. Okay, so I don't have time to write up the paper, but send a report to central government. The government eventually believe the SARS they did come from those live animal. So they start take action. I reported the central government on the January 2nd, year 2004. we have a meeting and conduct consensus to remove those animals. So they have a culling action since January 5th. Take about 1 week. Those red dot is representing the case. In the second outbreak of SARS, we only have 5 human cases. Each of them only be exposing by themselves. Those SARS cases cannot transmit to other people. So after culling, remove those white animal. The last case came out on January 7, and afterwards we have a peacefully world, no SARS anymore. Okay, so in Shanghai, SARS was completely under control. Okay, so we call this outbreak and avoid the SARS outbreak. So afterwards, we also conduct some samplings in different type of bats because our colleague found the SARS-like virus in bats. We conduct the surveillance, check a few thousands of bats. What we found, actually in different type of bats, they're holding a wide diversity of coronavirus than any other mammal species. We also found the bat coronavirus are older than other antivirus we observed in mammalian species, other mammalian species. So as early as in year 2007, we proposed that the bats may be the natural reservoir for all mammalian coronavirus. Okay, but this takes time to confirm. Year 2012, a patient from Qatar visited the UK to see doctors was identified caused by a new type of coronavirus. WHO named it MERS, Middle East Severe Respiratory Syndrome. Then it takes 2 years. Many scientists visited the Middle East. What they found, serologically, seemed the camel has a possibility. It's the intermediate host to pass this virus to human. But the local people, the Saudi Arabian and Middle East people, they don't believe, because what do they say? The camel stay with us for a few thousand years. Camel is their friend, their family member, and also their meat. So I visited Saudi in the year 2004. We conduct very systematically surveillance between the year 2014 and the year 2015. In total, we sampled 1,300 camel. Among these camel, about 12% of camel have the MERS virus. That means MERS virus did exist and endemic in the camel in the Middle East country. Genetically, this virus emerging and evolution in the camel. Camel is the major host for MERS virus. Okay. And they even happened recommendations to change to a new lineage circulating. So in the year 2015, when the South Korea has an outbreak, and the Laureate has outbreaks. This red dot is the virus we isolated. All of virus is surrounded both the Korea and Laureate outbreak, human virus. So this is the first time in the world provide solid evidence. The MERS did come from a camel. Okay, so I give them suggestion. Leaving those camel away, don't contact. Also give the suggestion to the doctor and the government. Any patient, if it's with severe pneumonia, you got to ask whether they have camel contact history. If that is the case, this patient should be isolated or quarantined. So since then, even the MERS virus still in the camel population until today, but no more large hospital outbreak. Also, we don't have pandemic. This virus, actually MERS, is caused about 30% mortality rate. It's much severe than currently ongoing COVID-19. Now, we come to the last questions. And the ongoing pandemic, SARS-2 or COVID-19 virus, how this virus emerged is definitely not from outer space. They only have 3 possibilities, at least here. Naturally emerged from animal, like the SARS, like the MERS. We did identify the animal origin, the human direct infection source. But why not this time? Second, the virus escape or leaking from some lab. This is the second possibility. The third one is the worst one, is the bioweapon attack or bioterrorism. So for the natural emerging pathway, there is some evidence support it. Because the bat SARS-2 related coronavirus were repeatedly detected in South China and the Southeast Asia country. Okay. We also detected those viruses from smuggling the penguins. These penguins are from Malaysia, okay, not from our country. So this smuggling penguin can come to our Guangdong region. It's also possible to go to Wuhan. So this is the support point that virus may jump to human. But why so successfully? We don't know. But there were other evidence don't support this. Our team conducted independent surveillance in the early 2020 in the Zhejiang Delta region. We sampled 14 mammalian species, more than 120 animals. We did find some alpha or beta family coronavirus, but no SARS-2. We also sampled another 8 non-mammalian species animal, and all of them are negative for SARS-2. So until today, right now, we failed to identify intermediate host or the human infection source animal. This question is still pending to answer. What's the remaining two possibilities? I don't want to give too much comment about this. Left it, these two questions for the task force team of WHO. I heard the second task force is nearly start work. Which then successful to identify, to answer this question. But as a scientist, I remind all of our audience, for science, nothing is impossible, or impossible is nothing. So we are still keeping an eye on this issue. Finally, I need to thank you, my big team, colleagues, the long-term effort, day by day, week by week, month by month, year by year, for the last more than 2 decades to make this story happen, to avoid so many damage from a virus. Okay. We do have a very good facility to conduct our work. Okay. Thank you for your attention.